Go like. Good morning and welcome to Chai and Why. Third Sunday of the month, we are at Prithvi Theatre with a very intriguing topic. However, before we start the session, in case you're joining in for the first time, let me tell you that Chai and Why happens on the first Sunday of every month at Prithvi Theatre, on the third Sunday of every month here at Ruparel College, and if a month has the fifth Sunday, we are live from a lab in TIFR. Today, it's a pleasure to introduce Professor Shyamalava Mazumdar with us. Shyamalava is sort of the expert in an area called bioinorganic chemistry, and he's been at TIFR for just 38 years and is a senior professor there, so he knows everything about this. I will let him introduce himself anymore if he wants, but more than that, he just likes to teach and has a lot of experience. Please ask a lot of questions. Online audience, please put your questions in the chat box so that we can monitor it and look at it. Also, let me say, if you're watching online, today the college is also hosting not just Chai and Why, but the UPSC exam, because of which we do not have the usual internet connection over here. We are trying this with a phone connection, though there are some jammers installed in the next room. So it might be a bit flaky. Let's hope for the best. With that, let me go back and share the screen again.
Done.
Okay, we are done. You're all set.
Okay. So let me start by telling that I like to teach, but I don't know how good I am a teacher. So that is the task which you have to decide. I have in TIFR a small team of quote unquote investigators who are actually working on certain things on exciting what is called chemistry. You can look at the spelling. Today, of course, I cannot talk to you about all we did or all we have been doing. We'll talk about something which is related to cyanide killing, and that's a story of a protein, we call it enzyme, which has iron in it. That itself is a oxymoron. That means, the biological system is known to have organic chemistry, organic molecules. But there is an iron there. So that itself starts from a very interesting thing. So I will be, this is my sort of plan of the talk today. I'll talk about cyanide. What is the use of cyanide? Why do we need if it is poison, right? Why do people make it? Then I will talk to you about different ways cyanide are obtained, okay? And then why it is toxic, what are the type of toxicity we have. Then we'll go into deep on what we do on our research, giving some idea about molecular origin of cyanide. And at the end, I will try to tell you something about how to remove the toxicity, basically antidotes of this cyanide. So I'm sure that you know that cyanide is poison. There are lots of stories about cyanide poison. Okay, story says that you don't know whether it is sweet or sour or salty, right? So somebody took it and started to write. It can only write S, and then it would stop. So cyanide is one of the very, very fast and notorious poison. The story of poison goes, prehistoric days. The earlier days, the kings used to make poisons and store it to, kill their enemies. And of course, cyanide had a competitor. Arsenic was very, very well known as a poison, but I'm not going to talk to you about it. Another day, we'll talk about arsenic. Cyanide is known as a poison for various reasons. It is highly water soluble, so it can act extremely fast. It is possibly one of the quickest poison known, and it can be the gaseous form, which is called hydrocyanic acid. It also can be, water soluble, very simple, colorless a white color powder, which is called potassium cyanide or sodium cyanide. They're like salt. Okay. There are stories about cyanide poisoning. In fact, one of them, which I have just taken out, I don't know whether any of you know about the story of Rasputin. Do you know? You can raise your hand. So Rasputin was one of the sort of, Russian monk, and he became a terror to people, and then people tried to kill him. The story goes that, if he was so powerful, you cannot kill him easily. Okay. So they tried to kill him by inviting him and give him a cup of tea and cake, which is laced with cyanide. But he had some way of knowing, and he could find the antidote of cyanide, and he could not be killed by cyanide. Later on, of course, he was killed by normal way. It was shot and he was killed. So cyanide poisoning is there everywhere. There is another interesting thing I can tell you. This ammonia synthesis, which was done by from nitrogen and hydrogen, is a very well-known industrial method called Haber process. Okay, and the man who discovered his name is Fritz Haber, and he got Nobel Prize also. But during the First World War, and he's a German, he somehow was supporter of the German group, and he actually discovered chlorine gas to kill people. Okay, and that was bad. Of course, later on he transformed himself, and during the Second World War, he being a Jew, he was thrown out of Germany, and he went to US and all that. But during Second World War, Hitler took over the lab in which he actually made chlorine gas, and used that lab to make a mixture called Zyklon B, which is chlorine and cyanide mixture. And that was one of the mixture which was sort of sold in small bottles, and they used to call it as gift case. Okay, and it's a poison gas, and that was used in concentration camp and many other places. So it has a very bad use in that sense. And later on in 80s, during the Cold War, it also was used in various places. The story says that even Hitler and his wife killed themselves by cyanide poisoning. Okay, and in recent, there are many stories about cyanide capsules. If some terrorist or some freedom fighter get caught, they take cyanide capsules and all that. So this is the story of cyanide. Now, let me just tell you that cyanide is not only poison, it is useful. And because of that, we, in our country, we use, globally, cyanide is used huge amount, 1.5 million tons per year. Okay. And there are chemical used. Okay. This is one of the chemical, those of you who might have, I have little bit of chemistry here, organic chemistry, organic compounds we use from toothpaste to, everything is organic chemistry. So making the organic compounds is one of the major tasks of chemical industries. And to increase the carbon length, let's say if you want to make from ethane to propane to butane or something of that type, you need to, the best chemistry which is available is using cyanide. And today, cyanide is required for making, many polymers. For example, perspex, this is one of the chemical methacrylate, which is synthesized using cyanide and butadiene. There is another chemical, and that is used to polymerize to make a plastic which is transparent. Also, the most popular and most well-known polymer, nylon, is also made from a cyanide compound, and that requires. So cyanide has a huge amount of application. Cyanide also is used for making, pesticides, then for fumigation of, against mosquitoes and other things. Okay, and also it is used for making dyes and pigments. There is one dye which is very, very well known. Everybody knows about it. It's a blue color, Prussian blue. Okay, So this is the structure of that, which is called ferric ferrocyanide. If you take potassium chloride and add cyanide to it, you get a blue color. Potassium chloride has colorless. Cyanide also is, potassium cyanide or sodium cyanide is colorless. And you take ferric chloride to that, the moment you mix them together, the solution will become blue color. And from that you can make Prussian blue, which is a blue dye, okay, which has, of course, some other applications also. And the chemistry of cyanide in most of the organic chemistry type is it takes a ketone, acetone type of molecule to which cyanide binds and forms what is called cyanohydrin and that polymers. So this is something which I don't want to go into detail. So other major application of cyanide is in gold industry. Gold has a very, gold is a inert, a noble metal, right? It's not inert, it's a noble metal. So it doesn't react with most of the things. So what cyanide is needed is to solubilize gold, okay? And this is the chemistry happens where you take gold, treat it with sodium cyanide, and it forms a complex of gold, okay? So that it is soluble in water. And that is actually an industrial process, okay, which is sort of used all over the world to get gold. And gold is one of the most important, commercially important metals. So cyanide, without cyanide, you cannot do that. So now how do we make cyanide? So much as use of cyanide. So major part of the cyanide, of course, is synthesized in industry and by a chemical method where we use ammonia and alkane, methane mostly, okay, in industry at very high temperature, pressure using a platinum catalyst. This is a scheme which is taken from Johnson Matthey laboratories, okay, and 90% of cyanide today is synthesized using this method, industrial. And once you have HCN, which is prussic acid, you can treat it with sodium hydroxide, you get sodium cyanide. You treat it with potassium hydroxide, you get potassium hydroxide, potassium cyanide, and things of that type. So, and those are also used because prussic acid, HCN, is highly volatile liquid. Okay, it has a light yellow color, and it's very, very dangerous to handle. On the other hand, potassium cyanide is although it is toxic, but you can keep it in a sealed bottle and carry it and things of that type. There are some, actually nature also has made cyanide, okay? And there are many organisms, mostly plant-based things, they actually make cyanide for their own protection, okay? And that's not very small amount. There are species which has very, very high quantity, that means almost like 600 ppm level concentration of cyanide. But it's not actually cyanide. Cyanide is poison for everybody. So they make cyanide in a way that there is a compound which they make, which is called cyanogenic glycoside. That means it's a glucose derivative of cyanide, and it stores it. So any, for example, a plant which has this type of cyanide sources, now if a grazing animal eats that plant, the animal has some enzymes, they will, hydrolyze, they will break down that glucose containing cyanide compound, okay, which is called cyanogenic glycoside, and that will produce cyanide. So, nature has developed a scheme so that, the tree will not die, the plant will not die, but the person or the species which is going to destroy them, eat them, they will die, okay? So,
is there any way to do the poisonous nature of HCN?
There are ways. I'll come back to that later on. I'll come back to that later on because it's already there. Okay. Now, some of the examples I have given. This is called limarine is the name of the chemical, linamarin, and it is found in some of the things which we know. For example, flaxseed, and flaxseed is also said to be very, very good. good for health, good for its omega-3 fatty acid and all that. So it's good for health, but we do have cyanide in that. Okay, so we have to be careful about it. I will tell you what care you have to take. We also have cyanide in seeds of apricot, which I think many of you know, in bitter almond, apple seeds, plums and peaches, and also cherries. Okay, and these are not present as the cyanide themselves, as I mentioned to you. These are present as a glucose derivative. You know, this ring which I am showing here is the CN cyanide. CN is the cyanide. Yes, I should have told that. CN is the cyanide. And this ring is the glucose ring, and which is connected to a carbon cyanide and something else. And if you remember that reaction of cyanide in organic chemistry synthesis, exactly same type of chemistry is done by the nature to make these molecules. Okay, cyanohydrin they call it. So this limarin, amygdalin, and there is another one called dhurin. Now dhurin is found in jowari. Okay, those are some of those type of, mainly depending on the type of plant, the concentration of cyanide changes. But cyanide is present in all these type of forms. They themselves are not toxic. So the plant, or say for example the apricot tree, does not die of cyanide because of this. Otherwise cyanide is poison for them also. Even for trees, cyanide is poison. Okay, it's not only for humans, it's for everybody. I will tell you why and how. But this compound of glucose containing cyanide will get digested in our body. Everybody has that enzyme for digesting it, and it will produce cyanide. Nature has designed in that way. So that's something very, very interesting. And the type of digestion process happens is this. As I mentioned to you, these are all called cyanohydrin. And when you use this enzyme, it actually produces glucose, acetone, and hydrocyanic acid. And that hydrocyanic acid is the poison. Okay, so this type of chemistry we can have in our gut, or in animals who have this, what they call it, gizzards, right? Birds, and where they actually digest their food, this enzyme is produced very, very large quantity in those places. So let me now tell you toxicity of cyanide. Okay, cyanide is toxic, right? How much toxic it is. So for humans, there can be toxicity of cyanide from fumes, something very, we don't know. For example, these plastics, this plastic, as I mentioned to you, they are, synthesis of them use cyanide. So there are cyanide containing groups are there. So if there is a fire, this fume which is produced by burning of plastics, they're not 100% burning, they're not producing carbon dioxide and water, and that produces HCN gas. So one has to be extremely careful when you are in a fume which has the plastic burning, whether it is a what you call this, motherboard, plastic boards, or plastic chairs or plastic furnitures. Everything will, on burning, produces cyanide, and that's not very small quantity. It depends on which plastic it is. For example, nylon has a very large quantity of cyanide containing compounds. Melamine, this melamine plates are used. Melamine, if you burn them, you get a smell, and that is poisonous. That has cyanide. And of course, this seeds of cherry, apple, even custard apple, sitafal, okay, apricot, okay, they contain this cyanogenic glycoside. Yeah. And actually that gives a special type of taste to little bitter type of taste. And the wild apricot, wild almond had a large quantity of the cyanogenic glycoside, but something happened. Nobody knows how exactly it happened. The natural modification happened in those almond trees, And the almonds which started coming out, which are being used, they have very, very low concentration of cyanide. It's not exactly cyanide. I should, clarify again and again. It's a cyanide-containing compound which produces cyanide. So if you need really to get the poison somebody by eating almond, you have to give him kgs of cyanide almond to get poisoned. So it generally doesn't happen. But main poisoning happens from the seeds of apple. There are some of these things can actually contain very high concentration. Okay. Of course, the other sources of cyanide is those who are working in cyanide industries, for example, gold industry and all that. That can have an accident, and there are cyanide is stocked in places, and where there can be accidents, cyanide can actually get leaked out and all that. And HCN is gas. It's a liquid with a low boiling, so it's a gaseous. And because of that, HCN is more poisonous than potassium cyanide, which has to go through the blood, and then HCN also goes through the blood, but it can actually get in much easily. Okay, there is another chemical called cyanogen bromide, which is actually used for the gas chambers. That is so permeable that even through the skin it can go through. So but ultimate chemistry is that cyanide is produced and it gets into the blood. That's the way it gets sort of. But the important point is, we don't need to worry so long as we eat within the limits. For example, of course, we should not eat apple seeds just like that. But if by mistake, if somebody swallows one seed of apple, doesn't need to go to hospital or something like that. That way, everything is poisonous. Only thing, small quantity of poisons can be medicines actually. Okay, that's what is also done. Possibly that has some application. I had read somewhere that apricot seeds, they have some health benefits, although they have large quantity of cyanide also. So they might be actually used to kill some of those parasites in our body, and that helps. Now let me just tell you what is the symptom of cyanide poison. How do you know that somebody has? In fact, this happens more for cattle or birds. If they eat, for example, if cattle is grazing in a field which has the things like Sudan grass or jowar, some special type of jowar has a very large amount of cyanide, and if some of these cattle are eating them, that can actually kill them, and that is the thing which happens quite often. And the problem happens is weakness. They fall down, and there is a nausea, confusion, headache, difficulty of breathing, seizure, and a loss of consciousness, and finally heart attack. Okay, that's what happens. And cyanide poisoning actually also depends on the type of dose. So, for example, if you take very, very small amount of cyanide, that may not cause any visible damage to the system. But if somebody takes a large quantity, that can actually damage the life process itself. So cyanide poisoning, you can have an acute poisoning where nothing can be done. If somebody by mistake swallows potassium cyanide, or gets into a place where there is a huge amount of HCN gas, then the person will have a seizure, will not be able to breathe because the oxygen is not being used, and heart attack. They lose consciousness. But also, if somebody is taking a low amount of cyanide, mostly those which are from natural sources, like if somebody is having a bitter almond, okay, regularly, then slowly this problem happens, and it's not that it cannot be really taken care of. So somebody who has taken this acute cyanide is, once somebody has consumed cyanide, nothing can be done. But this chronic cases of cyanide poisoning can be actually taken care of, which I'll talk to you later on. And the symptoms are headache, drowsiness, and also sometimes vomiting, vertigo, and things of that type. And there can be also one interesting thing happens if somebody has a cyanide. If one tests the blood becomes even more red. Okay, I'll tell you why it happens. The blood oxygen concentration increases in cyanide person, which is unlike in many other cases. So all these things tells me that it has something to do with the blood, right? So oxygen chemistry and the blood is important for cyanide poisoning. So we know that respiration is important, and blood does the major job of blood is respiration. So we take in oxygen, we take in air, and oxygen goes into the blood. Okay, and which molecule carries the oxygen? Hemoglobin, right? So hemoglobin, and we have lot. The blood is carrying oxygen through hemoglobin, and the red color of blood is actually from hemoglobin. Everybody knows it, right? Is anybody who doesn't know this? Everybody knows, right? So hemoglobin is the first thing. So let me just give you some brief discussion of heme. Heme itself is a very interesting molecule. We are chemists, so we know that this is a very, very nice looking iron containing molecule. Okay, and it's a, what is it called, or aromatic molecule. And this person by name Hans Fischer, he actually got Nobel Prize by making this molecule in chemically. Okay, and it is present in many type of proteins, a large number of proteins. So it is so those proteins which has heme inside that, they are called heme protein. So there are proteins which has the heme. Also there are various types of heme. I don't want to go into detail of that. So there are heme present in, for example, this one. Yes, they are largely, heme means they have iron. But this is another interesting, sidestepping. This ring type of molecule which you can see, which is called porphyrin. This is the basic structure which somehow nature has liked. And if you see chlorophyll in color, molecule which is very similar to this, but that's not iron content. That has magnesium. So that has a completely different type of function, but the molecule is very similar. There is another molecule called vitamin B12. That also has a very similar type of structure, but not iron, but it has a cobalt. So I'm talking about heme. That means these are the iron complex of the porphyrin ring, but that also has so many variations. You can have a heme B, which is having some small variations, is present in hemoglobin. Okay. Heme C is another one which has some other small variations in the structure, is present in another protein called cytochrome C. That's also a very small protein, but there's a very, very wonderful function it does. Heme A is the one which is present in the protein which I'm going to talk to you today, and heme D is present somewhere else. And slight variations or same type of heme can actually do different type of function in depending on which protein it is sitting inside. Okay. For example, oxygen transport and storage is done by hemoglobin and myoglobin, and that has heme. There's a heme which does the job, and the protein part is just holding it. Okay. Similarly, there are useful molecules like nitric oxide and carbon monoxide, which are also there in our body. They are synthesized, and they have very, very important job. They're very small amount. And there are protein called nitrocorin which does that. And the cytochromes, as I just mentioned to you, they actually do a very important job called electron transfer. I don't want to go into detail of that, but this is something which is the most important and key function of respiration. And of course, catalysis, many of you know, the catalysis for catalyzing reaction. That means it makes some reactions easy and some products being formed. And there are many of these enzymes which they are called enzymes like peroxidases. They actually destroy hydrogen peroxide from our body. Okay, things of the catalysis, they destroy hydrogen peroxide and superoxides from our body, which are detoxification enzymes. But hemoglobin is the major one. But if you look at the hemoglobin structure, hemoglobin is a red type of molecule packed with a protein called hemoglobin. These are the RBC, sorry. And each of the hemoglobin molecule has 4 of iron porphyrin, 4 hemes. But if we look at this, the iron porphyrin molecule which holds oxygen, when oxygen binds, oxygen goes and sits to iron on top of iron. Okay, but the iron is in the iron 2+. See, iron, see this in chemistry, these metal ions can have different oxidation state. Okay, they get oxidized. Ferric chloride is iron 3. Okay, on the other hand, ferrous sulfate, mostly we take ferrous salts as iron supplements, ferrous citrate or ferrous sulfate. Those are the ones which iron tablet will have. If you look at those who are taking iron tablet, you will see there is a ferrous salt. Or ferric chloride, ferric oxide, they are there in rust, for example. In iron rod, so these oxidation state differences are very, very important. Whole chemistry of the metal changes. For example, metallic iron will not bind oxygen, right? Iron rod will never bind oxygen. So you have to have an iron 2+ to bind oxygen. But iron 2+ will bind oxygen, but it will not, and it also binds carbon monoxide. And that's why carbon monoxide poisoning of hemoglobin is something which is very, very, widespread, especially in villages. Respiratory strain, because it stops breathing and things of that type. Somehow it does not you bound form in your blood, which is not normal, because consumption of oxygen from hemoglobin decreases if somebody has cyanide poisoning. So now comes to what is the molecular origin. So what might happen? It is not attacking the hemoglobin. So it attacks an organelle in the cell called mitochondria. And this mitochondria, as you know, is the cell's powerhouse. That's the place where energy is generated. Whatever food we eat, ultimately it produces energy, and that energy production happens in the mitochondria. So let me just go a little bit deep into it. So this is what we call the cellular respiration. The first one within the respiration process, which is external respiration. But when you do the cellular respiration, that inside the cell, what happens? Has membranes. Outside membrane, but inside, but it doesn't actually act as a membrane separating something, but this membrane itself of a large number of enzymes, which actually convert various things, okay, or catalyze certain reactions. Those are called membrane proteins. And these guys. The problem of cyanide poisoning happens because one protein particularly called cytochrome C oxidase, which is bound in the membrane of mitochondria, stops functioning because of cyanide. I'll tell you why it happens and how it happens, but cytochrome C oxidase, this is the most important, according to me, and many people believe that this is the most important enzyme for our survival. Okay, and as you know, as I just now mentioned, that mitochondria is the one which is powerhouse, and the cell mitochondria, the efficiency of production of energy, that means ATP, depends on how many cytochrome oxidase and how many respiratory chain proteins are present in the mitochondria of the person. Mitochondria itself also has DNA which produces this, so it doesn't depend on the actual cellular DNA. This has something else to talk to. As you know that we are born by mixture of the genes of father and mother, right? and mother cell is used. So the mitochondria, what we have got is our mother's mitochondria. Okay, and the mitochondrial DNA comes from mother. So if somebody has a mother strong, the children will be strong. Okay, because more energy can be produced in the body. Second important thing is the mitochondria is present in all living organisms which uses oxygen to survive, which are called aerobic life. Okay, without oxygen we cannot live, right? And that happens to not only us, it happens even for some bacteria. So all through we have mitochondria. I'll just tell you in a moment. Okay, before that, let me just tell you, these are the proteins. I'm just showing as these are initially they were given names as complex 1, complex 2, complex 3, complex 4, like this. And later on, because these mitochondrial proteins are very difficult to crystallize and do crystal structure, and complex 4, where you can see the oxygen is getting converted to water, this is the one which is later on has been named as cytochrome C oxidase, because that's the function, and it takes this cytochrome C, goes and binds here, and that is why they call it cytochrome C oxidase. So before I go into more of this is I'll talk about some proteins, and proteins are made up of amino acids. So I'll just briefly tell you, these are some of the amino acids. Amino acids are nothing but small molecular fragments having ammonia component and the acetic acid component. Okay, so it has property of ammonia and also property of acetic acid, and they are polymers. Okay, and there are some amino acids which has some extra groups. Like for example, outside the square bracket, I'm showing some of them, like cysteine has sulfur, aspartic acid ASP has some carboxylic acid group, which is like acetic acid. Okay, and then histidine has a small 5-membered ring containing nitrogen outside the square bracket. And the square bracket is the one which is the amino acid component which polymerizes. And those side chains are the important for anchoring different metal ions, okay, which is called coordination chemistry, which I'll go into more detail later on. And there are many other rules. This, for example, if you have a aspartic acid which is a negatively charged, okay, negatively charged means this side chain carboxylic acid group is the one which is giving negative charge. Then on the other hand, if you look at lysine, LYS, it has a nitrogen, which is an ammonia type of molecule, and that gives a positive charge. So that's how the different charges, different type of chemistry comes from these different amino acids. And these proteins, which are made polymer of amino acids, they can take different shapes, like for example, helix, you can have a beta sheet, depending on the side chains, which has carboxylic acid, amine groups, and things of that type. They come close together. If there is an amine and carboxylic acid, they can come close together and interact. And that's how they take different type of shapes. It can have actually 3-dimensional shape. Okay, there can be cavities inside and all this, and they are very, very precise and highly stable structures. And that is the story of the protein. You can have protein as simple as having maybe 80 or 40 amino acids, okay, polymer, 100 amino acids, 1000 amino acids, and they are sort of forming a special type of structure, some part of helix, some part of beta sheet having some bent and all that. You also can have protein which has this type of polypeptide chain, multiple of them. They call subunits. So one subunit is here, another subunit is just physically associated to each other. And that makes a much more complicated structure. And that structure can be actually bound to a membrane. And membrane is, like, for example, mitochondrial membrane, which I mentioned to you, is highly hydrophobic. That means they are oily. So this, protein, if they are sort of bound to a membrane, they remain stuck to that. They don't get, separated out. And that makes a complex, protein complex, they call it. So this is a small protein called cytochrome C. I mentioned to you, the cytochrome C is also a heme protein. It's one of the smallest proteins, and its job is to do electron transfer. So what it has a heme, and this is the heme slightly different. As you can see, heme is actually bound to the protein. Some of the amino acids are bound actually to the heme. And iron is in a +3 oxidation state. And it accepts electron to become +2. Okay. In the electron transfer chain, it accepts electron from one place, becomes +2, moves, goes and binds on cytochrome C oxidase, and gives electron. So it shuttles electron. Its job is just to take electron from one place, gives electron to another one. That means it itself gets reduced, goes, reduce something else in cytochrome oxidase, and itself gets reoxidized and again comes back. So it's a shuttling job. But that's one of the key molecules for respiration. Without this, respiration will stop. But you can see that this has iron. Generally, those who have some exposure about coordination chemistry, iron like to bind to 6 atoms. They call it ligand. And iron coordination site is full. So it doesn't actually attack cyanide. Okay. The one which binds cyanide is called cytochrome C oxidase. And that's the enzyme which catalyzes this reaction of converter of oxygen to water. So this is the catalytic reaction. This is a chemical reaction which catalyzed by cytochrome C oxidase. And this is the structure. I don't want to go deep into it, but I will show you this structure again and again. But that's why I gave you some introduction that the protein structure is like this, and there are side chains which are stabilizing it. And this, in this case, different colors are shown because they are different polypeptide chain, they are different polymers, and they are assembled together because they are actually embedded inside this membrane, which is an oily structure. Inside that, it is stable. And one more small thing I'll tell you, this crystal structure of this was discovered in 1995 from both bacterial source and from mammalian source, human. It was actually, the first one was reported from bovine, from the cow's heart. And it was found that, of course, the mammalian one, the bovine heart, that is much larger, has 13 subunits. But bacterial one has 4 subunits, 4 proteins assembled together. But those 4 proteins in bacteria are identical to the 13 ones which are found in cow. What does it tell? It tells that the bacteria and the cow actually are the same. They have a sort of, they have come continuously. Their only proteins have been added, but the bacterial proteins are still retained. And these proteins are synthesized from the DNA, which is found in. So out of these 4 subunits, the first 2 subunits, which people have now understood their functions. So I will talk only about the subunit 1 and subunit 2. And they are the ones which are very key and fundamental things about the function of this enzyme. So I don't subunit 2, which has a head type of thing, and there are some parts which is going inside the membrane, and here you have a copper containing center. So this also does not bind cyanide, but it has a very interesting copper compound, which is a purple color. Now, subunit 1, which is completely buried inside the membrane, has more than one. If you look at it, you see it has 2 heme centers. Okay, and that's the one which crystal structure is very, very difficult, and it was solved, and it found that it has a heme A, which has a iron is bound to the porphyrin ring, plus there are 2 histidine amino acids. So that is not a candidate for binding of cyanide. The second one is heme A3 copper B. That means it has the heme center and there is another copper center. And this is the center which is the culprit. That's the center which has, there are lots of, Nobel Prize has actually been given to the discoverer of this, that this is the center where oxygen gets converted to water, and this is the center where cyanide poisoning happens. So I will very briefly tell you that the centers looks like this. So there are copper centers, then the heme A3 copper B. These are the 3 metal centers which are functionally important. There are many other metals which are present. And the cytochrome C, which is shown by separation, that gives electron to the copper center, copper A, and the copper A gives electron to the iron of heme A, and that goes to heme A3 copper B center. So that's how the electron transfer pathway shows. It's a very complicated. I don't want everybody to understand it. Each of the steps is one paper. Okay. And if we really look at it, this is the center where you have a heme and there is a copper, which is called copper B, and the oxygen actually bridges between them. That's the intermediate which has been discovered, identified. And it forms, and then interesting thing happens. The oxygen, in case of hemoglobin, what happens? Oxygen binds, and then hemoglobin molecule moves, and it again releases oxygen to somewhere else. So that's how oxygen transport happens. In case of cytochrome C oxidase, inside these is the oxygen binds, but it gets transformed and it produces water. And that's the very complicated thing. I don't want to tell you details, but basic thing happens that electron transfer takes place across the membrane. Okay, so this is the yellow box schematically shows the membrane, and the electron moves across the membrane to the heme A3 copper A center where oxygen gets converted. So how exactly it happens is something still people are trying to understand. But one thing is known, that there are 4 electrons are required for getting 1 oxygen converted to water. And for this process, cytochrome C is the person, is the molecule which gives electron. And I have shown cytochrome C, how does it take electron? It is iron free. It takes 1 electron, becomes iron 2, goes and gives that electron. So it gives 1 electron at a time. So 4 electrons, that means 4 cytochrome C has to settle. The first electron it gives, it goes to the copper A center, then from there it goes to heme A, then from there it goes to heme A3 copper B, and second electron again comes like that. First electron, second electron. Now if you look at it, and this you have formed this iron oxygen copper center in the active center. Now oxygen molecule, if you look at it, if you give 1 electron to it, that forms, do you know what it forms? Superoxide, O2 minus. If you give 2 electrons to oxygen, it forms peroxide. So as you know, superoxide and peroxides are poison. They are highly oxidizing things. They are called reactive oxygen species. Okay, and those things are bad for us, bad for our health. Okay, because it's damage to the cell. So cytochrome oxidase function has attracted people so much, because when it functions, when it converts oxygen into water, it does not release any of those superoxide or peroxide. So it has a very nicely evolved mechanism nature has developed, so that it can actually bind in the species A, it's a iron bound to oxygen and copper in between. But then there are lots of complicated steps happen, and this oxygen-oxygen bond is broken by this enzyme. Okay, and it releases 2 water molecules, you can see in 2 steps, and takes electron 1 at a time. So this process is extremely important for understanding of the function of this. But that's not the everything. So what it does actually, when it does this conversion of oxygen into water, it parallelly pumps proton across the membrane. So if you look at the reaction, which I have shown here, somewhere else, okay. 4 electrons and 8 protons are given to it. And these 8 protons are actually given from one side of the membrane. And it uses 4 protons to produce 2 water molecules, and the 2 protons it pumps out of the membrane. So proton is positively charged. So at the end of this respiration, what happens? Some protons are pumped out. So outside the membrane becomes positively charged, and inside becomes negatively charged. So that becomes like a battery. If 2 charges are separated. And in fact, scientists started dreaming that we can possibly make this enzyme, run it, and produce a battery. So that will be one of the smallest battery which it can be made. Okay. So of course, that research is still going on. This is not the topic of today's discussion. But take-home message is that this oxygen gets converted to water without forming superoxide and peroxide by cytochrome oxidase. But in some time it does happen when the cytochrome oxidase is not able to function properly. without the cyanide poisoning even. How it can happen? It can happen by aging. For example, brain cells, they don't die. We have the same number of or less number of brain cells than what we were born with, right? So the cells become old, the membrane becomes weak, and if somebody is drinking or there are other type of things which can cause the damage to the membrane, and then the assembly of the subunits of cytochrome oxidase gets distorted, and that produces a slip in the catalytic cycle, and it starts releasing hydrogen peroxide and superoxides, which are poison for the cell, and that's why Alzheimer's disease, Parkinson's disease, etc. starts forming. Less polar means oily or which are polarity means like water is highly polar, alcohol is less polar, okay, less polar. Oil is non-polar. So too much oil actually can create problems. That's what doctors suggest if somebody has a heart problem, take less oil, because oil will get accumulated in the membrane and that can damage the membrane structure. And cytochrome oxidase function can get distorted. So we learned few things. What we learned that cytochrome oxidase is a membrane-bound protein, and the oxygen goes in and binds ultimately to the heme A3 copper A center, copper B center, as a bridging between the iron and copper, and then by series of electron transfer, that oxygen gets converted into 2 molecules of water, and they are released. That's how the whole function happens. And while doing that, it actually pumps protons. So for 1 oxygen molecule, 4 protons are pumped across the membrane. So that produces a positive charge outside the membrane, which is called, you can have a charge gradient like a battery. And this energy of the battery is actually used by another enzyme called ATP synthase, which stored this energy in the form of ATP. So ATP synthesis will stop if this enzyme stops functioning. Right? Understood? So now this is where the cyanide comes. It has been found cyanide goes and does not bind to the copper A center. It does not bind to the heme A center, but it goes and binds to the heme A3 copper A center. Can you see this round which I have circled? This is the crystal structure of cyanide-bound cytochrome oxidase. So it goes and binds there. And once it binds there, the cyanide cannot get converted into, it cannot be broken. Oxygen can be broken into water, but cyanide remains cyanide. It tightly binds, it kills the cell, kills the enzyme. So enzyme cannot function anymore. So the moment the cyanide is formed, what happens? This is the electron transfer route I have told you. Cytochrome C to copper A, then copper A to heme A, and heme A to heme A3 copper B, where the oxygen gets converted. The moment the cyanide binds, this step gets stopped. And if this step stops, the proton pumping gets stopped. Cytochrome oxidase cannot pump proton. Now, if it cannot pump proton, ATP cannot be synthesized. And if ATP cannot be synthesized, the cell will die. And if that happens in the heart or in the brain, we die. How much time it takes? Cyanide binding is extremely fast. So if somebody is having a pulse rate of 60, let's say, that means in 1 second, the whole body is having the same molecule going in. That's good enough. Okay. So it can actually go and stop some of the cytochrome oxidase molecule. And if it goes and stops some molecules in the brain or stops some molecule in the heart will stop functioning because heart doesn't have energy. And heart produces, this pumping of heart requires enormous amount of energy. Major of the energy is required here. Okay. And the next is in the brain. So if cytochrome oxidase is not functioning, then the cell will die. You will have so much oxygen in the body. Blood has a lot of oxygen, but oxygen is not able to utilize because its cyanide has blocked its entry. That's how cyanide poisoning happens. But there are some antidotes. And if somebody has very high amount of cyanide poisoning, okay, nothing can be done. But if somebody is having a slow poisoning of cyanide, for example, if somebody is ate a lot of bitter almond, okay, has started feeling dizzy and all sorts of things. Then what you have to do, that cyanide is there in the blood. So you have to stop that cyanide to go and bind to the cytochrome oxidase. For doing that, you need to give the molecule, give the body something which will start binding cyanide. Hemoglobin does not bind cyanide, but we have a lot of hemoglobin. So we can add some chemical like sodium nitrite or amyl nitrite and things of that type. They can actually damage hemoglobin. They will destroy hemoglobin and make it iron 3. So that hemoglobin molecule will not bind oxygen anymore. Okay. But if cyanide is there, it will bind strongly to cyanide. So that way you can actually remove the cyanide poisoning. Of course, you are damaging your body because your oxygen binding capacity decreases, but at least cyanide poisoning can be taken care of. Because hemoglobin always can be given from outside. Also, you can add a chemical called hydroxycobalamin, which is vitamin B12. As I mentioned to you, is a cobalt complex of the porphyrin, and that has actually cyanide. The vitamin B12 is the one which we take, okay, because stool or anything, is a cyanocobalamin. It's a cyanide complex. That's the medicine for us. Okay. So if you add the hydroxy complex of the cobalt porphyrin, which is called hydroxycobalamin, immediately it will very strongly bind cyanide, whatever is there in our body. So before the cyanide, if you have a small amount of cyanide, first it goes into the blood, and that can be actually destroyed. And if somebody has cyanide poisoning and but still not above the threshold, then you can use something for curing, and that is by an enzyme which we have in our body called rhodanase, and it actually uses sulfur containing compound and the cyanide and produces thiocyanate. Thiocyanate is harmless. Okay, so that sodium thiosulfate is a chemical which is called hypo, which is used quite often in some, I think, barber shop earlier days they used to use. Okay, so that can be, and also it is used in photography earlier days. So that can be actually given as a medicine, so that some amount, a small amount of cyanide can be converted to thiocyanate by our own body's enzyme. But it has a very long-term and not very efficient treatment. And especially these are used for cattle, those who have eaten a lot of, jowar or something, jowar plants, or in the grassland which has a lot of cyanide. They can be actually treated with this type of chemicals like sodium nitrite, sodium thiosulfate, and mixture of them. The medicine is available for them. And also it has been shown that garlic extract, it has a lot of sulfur, so it can actually go and bind to cyanide and convert them into thiocyanate. So that is the type of thing. This is the cobalamide structure which I talked about. So I end here. Just to summarize, I talked to you about what are the uses of cyanide. I told you the cyanide is an important thing. It's not only poison. It has a lot of application, good application. Even vitamin B12 needs cyanide. And sources of cyanide, I also talked to you about that. The industrial technique where you make hydrocyanic acid, which is required. Okay, and also the nature makes cyanide precursors, I should say. Now, cyanide toxicity, I mentioned to you that there are various sources of cyanide toxicity. It can be hydrocyanic acid, potassium cyanide. Also, it can be from the bitter almond and things of that type. But the effect of toxicity depends on the dose. It's not always going to be toxic. It needs some threshold amount. And I spent quite a lot of time on the molecular origin, discussed about respiratory chain, which is very, very important, how it is actually converting cytochrome electron, which we actually, eating produces electron, nothing else. Okay, So it's a food is nothing but to reduce something. It's like a fuel. Like fuel we burn. Food is something which we burn in our body to produce water. Oxygen is getting converted to water. That's the ultimate chemistry happens. And that is done by cytochrome oxidase, okay, converting oxygen and the electron. And it produces ATP. And then how exactly cytochrome cyanide goes and stops that reaction. And I just now mentioned briefly about the how to remove this toxicity, okay. Antidotes, okay. You cannot have an antibiotic or any other medicine against cyanide, okay. But these are the chemicals which can be used. You have to keep cytochrome oxidase available so that it can bind oxygen. That's its natural function. So if you add something else which will keep it blocked, then anyway the function will stop. So it will be like cyanide only. Okay. So that will not solve the problem. Okay. So this is our team. Dr. Muhammad Tahir is sitting here. His photograph is there, and some of the people who are not here anymore, they have finished their PhD and moved on, and some people are still here. Okay, they are working on it. They are investigating, trying to find out various issues, not only from cytochrome oxidase, other proteins we work on. Thank you very much.
Before we
Yes. The first question was, yeah.
The one question is, can the cyanide CN be broken down?
The CN, nitrogen and carbon has a triple bond. Okay, different bonds are there. So carbon nitrogen bond is extremely strong. It cannot be broken so easily. While that's why cyanide, like carbon monoxide, also is very, very, very stable molecule.
So it's basically like it's unbreakable, practically unbreakable.
It is practically unbreakable, chemically, yeah. Yeah.
So the next question is, how are there any ways that detect cyanide poison, and how do they?
Yeah. Actually, I briefly talked about the cyanide poisoning. There are symptoms are there which are very special. They will what they do, they attack the nervous system. Okay, you start feeling dizzy and things of that type, and there are many other poisoning can cause that, but increase of oxygen concentration in the blood is a symptom. That means the blood color becomes deep red, and that is a sort of a direct test. Mostly for cattle, that's how they detect cyanide poisoning.
Color,
visual inspection of the. It is not. I think all species which uses oxygen for survival will die on cyanide. Only those bacteria which does not need oxygen for survival, they can survive. In fact, I forgot to tell you one more thing, that this symptom of cyanide, once cyanide goes and blocks cytochrome oxidase, the oxygen consumption stops. So blood has a lot of oxygen, but system is required to have survive. So there is something called anaerobic respiration. So our body starts doing that, and anaerobic respiration produces lactic acid. So the acid concentration in the cells increase, and once acid concentration increase, you start feeling burning sensation. So hot flashes and all these things are another symptoms you can see.
There's one very long short question, but is it possible that perhaps
at least I'm not aware of anybody is using, but CN derivatives can be used for various other purposes. It's being used.
So That's all. So, okay, so for our online viewers, let's try. We'll be visiting TIFR, TIFR, be a virtual visit for the audience, but it will be live on the exact time. Okay, so we are ending on this session now, and if you can have some chai.
Yeah.
Then
We can have a discussion. Okay, so you can leave.
Yes.
The net connectivity was poor, right?